Hello, I'm Professor Brinton, and welcome to episode 15 of the Notecard Review. Today's topic is the different types of DNA polymerases. There are three main ones and a fourth that we've briefly discussed as well. For starters, DNA polymerase 1 is the enzyme that comes in and replaces RNA primers, which are shown here in green, highlighted by blue, that were originally added to match with the strand of DNA during replication. Okay, so this molecule is always going to be working in sync with ligase to create that missing bond that's there at the gap between what used to be an RNA primer and the initial DNA nucleotides. DNA polymerase 2, we only talked about very briefly in our discussion of nucleotide repair. Okay? So when nucleotides are mismatched or otherwise damaged, say from UV radiation, sometimes it can create errors in the sequence. A nuclease is going to be the enzyme that actually removes the problematic section, but DNA polymerase 2, the molecule here, shown in yellow, is the one that comes in and adds the correct nucleotides. Again, this one is going to be working in concert with DNA ligase as well as the nuclease in order to perform its function of replacing those erroneous nucleotides. DNA pol 3 is the one that we've actually discussed the most. So this is the workhorse molecule from DNA replication. It's the one that binds to the initial primers and creates DNA running from that 5' to 3' direction in the replication process. So this is going to be the predominant molecule acting on the leading strand, and it's going to be the molecule that's creating each of the different Okazaki fragments by adding those nucleotides in short sections there. Okay. This molecule is, in a way, somewhat more independent. It's going to be working in concert with the primase because it has to have a primer to attach and begin this process. The fourth is DNA polymerase alpha. So this one we discussed only very briefly as part of our discussion on telomeres, which is the extension of the end of DNA strands so that the information isn't lost in the repeated copying process. So DNA pol alpha is the one that comes in and attaches to the primer created by telomerases and fills in that backwards gap of DNA with that repeated section there at the end. So DNA polymerase 1, 2, 3, and alpha all do the same thing in that they are all adding DNA nucleotides to an existing strand, but they're all functioning in different parts of various processes surrounding DNA replication.